Okay, I guess we can start. So before I forget, I prepared the exam, so the exam will be in the same format as the one of the course of Mauro Pastore and Bruno Loureiro. So there will be 20 multiple choice questions, and you will have to answer without using notes or electronic devices, okay, of course. So I mean, these are simple questions on what I said in the lectures, and I think if you follow the lectures, it should be okay. Okay. So now to conclude the discussion, I wanted to finish the discussion on graphical models and then show you an application to proteins. So let's start by concluding the discussion on graphical models. So we are trying to infer a graphical model, which means a model, a probability distribution over our data, which is made by local terms, one per variable, plus interactions. And these interactions form a graph, where the graph is the set of pairs ij such that j is non-zero. So there is a direct interaction between the two variables. We have seen in the case of Gaussian variables that we can compute many things explicitly, and we can infer, we have an estimator for the matrix of the couplings, and we can regularize, and we can study what happens, how many data we need, and so on and so forth. But okay, in most applications you don't have Gaussian variables, you have more complicated things, and in particular you can have categorical variables. So now we move to the case of categorical variables, and for simplicity today, for this discussion, let's restrict to binary variables. So, for example, 0, 1, or -1, 1 if you prefer, but here it's 0, 1. So you can think to these binary variables, for example, as if you have recordings of neurons in a given bin, time bin, you can say 0 means that the neuron is not spiking, and 1 is the neuron is spiking. And you would like to infer these parameters. The hi will tell you what is the probability of neuron I being active, and the jij will tell you what is the coupling between neuron I and j, which is related to the probability of being both active. Okay. So from this, then you can infer, you can get information about your system. For example, which neurons are the most the strongest in interacting, and so on and so forth. So we choose this model, which is a Boltzmann distribution of an Ising model with local fields and two-body interactions. And we said we can motivate this choice in two ways. One, we can say, okay, we want to do maximum likelihood inference. So we choose a likelihood. So in this case, this will be our likelihood of the data, sigma. Sigma are the data. Given the parameters, h and j, I don't write it explicitly, but this will be the probability of sigma given the set of parameters h and j. And you can say, okay, I choose this likelihood because it's simple. It's the exponential of a Hamiltonian with one and two body interactions. And you could, if you want, add 3 body interactions, 4 body interactions, then you would have a more complicated graphical models and many more parameters. So to start with, we stop at the simplest non-trivial term. Of course, if we don't put this, then we have independent variables, and then we don't have information on interactions. So this is the simplest choice, if you want, for the likelihood of a graphical model. Or if you prefer, you can say we want to do maximum entropy. And so this would correspond to the maximum entropy distribution with the constraint that the expectation of sigma I on the data is equal to the expectation of sigma I on the model, and the same for sigma I, sigma j. So this is also the maximum entropy distribution that is consistent with fixing the firing rate of each neuron and the joint firing rate of each pair of neurons, if you think about neurons. Okay? I also told you yesterday that in these models you have an overparameterization if you write it in the most general way, because in principle if you have two states, here you should have a 2 by 2 matrix and here you should have two fields, but you can always remove one field on each side and one line and one column of the matrix. So if you have binary variables, you can reduce to this form with only one field and one coupling on each pair. Okay, so now the problem is we have data. How do we get the HI and the JIJ? So the first possibility we explored is Boltzmann learning. So Boltzmann learning is the exact way of getting the fields and the couplings in this context. It is exact in the sense that either if you do maximum likelihood or maximum entropy, in both cases, if you maximize the entropy or maximize the likelihood, you obtain that both quantities are concave, so they have a unique maximum. The maximum is obtained when the moment matching condition is met. So you have N fields, you have L fields and L times L minus 1 over 2 couplings, and here you have L equations and L times L minus 1 over 2 equations. So these equations fix everything, but in order to solve them, you can just do gradient ascent on the likelihood or on the entropy as you want, and this will give you iteration equations of the form: if your model has too low firing rate for neuron I, you increase the field so that you increase the firing rate. So this will give, in general, something like something like this. If your firing rate is too low, you increase the field. If it's too high, you decrease the field. And the same for the couplings. So these will converge to the unique maximum if you, of course, if you choose wisely your learning rate, of course, otherwise you can have convergence problems. And okay, here it's of course, it's the model at time t. So you will have couplings and fields that evolve in time, where time is the number of iterations of your algorithm. And after a long enough time, this will converge. The problem with Boltzmann learning, it is exact. So once you are at convergence, you have your probability distribution, and this is really the maximum entropy distribution. The problem, you have a certain number of problems. First of all, this is not a problem, this is an advantage. These two things, you compute them at the beginning of the algorithm, and that's it. So this is particularly good when you have many data. If you have many data, computing these things takes a bit of time, but you do it once, and then you are done. You have your L magnetizations, and you have your L times L minus 2 correlations, and you store them in memory, and you run, and it's okay. So when you have many data, this is an advantage. But the problems are that you have to compute these two quantities. And to compute these two quantities, as you know, you cannot compute magnetizations and correlations for an arbitrarily complicated Ising model. So you have to do Monte Carlo. So you will need Monte Carlo sampling to compute these things. And here you have a disordered Ising model, so you know that when you do Monte Carlo in a disordered system, you can have convergence problems of the Monte Carlo. So the Monte Carlo can be slow, and this can take time. And if you're not careful enough, you have bad estimates of your expectations in the model, and the Boltzmann learning has convergence problems. So there is a long series of studies of how to properly do the training, and there is a lot of progress in the last years in particular thanks to the work of the group of Beatrice Seoane. So they found various ways to improve the strategies to do Monte Carlo in a proper way. So you can check their work. It's very interesting. Now, the other problem is that also the Boltzmann learning itself, even if in principle it's guaranteed to converge, can have convergence problems in the sense that, okay, it's convex, but still it's a high-dimensional space, and a convex function in a high-dimensional space can still have very flat directions. So sometimes you can have also a bit of a slow convergence of the Boltzmann learning. So even if you have a good estimates of the moments of the model, this can take some time, and you have to be careful in the choice of the learning rate and so on and so forth. So overall, this is a good strategy, but okay, in the modern implementation that we have, it will require maybe 1 hour or a few hours on a GPU to have a good convergence. In a model with maybe 100, in a protein, for example, I will show you with 100 sites, it will be something like that. And then when you increase the size of your data, it becomes slower and slower. So you can have problems. So this is why people have explored alternatives, in particular in the past when computers were slower and there were also less data and so on. So there are other methods. We have seen the mean field approximation. So the mean field approximation is based on applying mean field to the easy model. And when you do that, what you get, we discussed yesterday, you get, okay, now let's call, just to simplify, let's call this the magnetizations, let's call the average over the model in this way. So the mean field equations give you an equation for the magnetization that has this form, that is obtained, as we discussed yesterday, from the conditional probabilities. And so here, the average of H is equal to Hi plus the sum over J of Jij sigma J average. So from this equation, you can take a derivative as we did yesterday, and you obtain that Jij is simply C minus 1 Ij for the minus C minus 1 for the off-diagonal elements when I is different from J. The diagonal elements of Jij, we suppose they are 0. We don't have a self-interaction because there is already the field. So, okay. So from this, you can say, okay, if I want to fix the moments, I want Cij to be equal to the Cij of the data. So I will put here Cij of the data, and I get Jij. And then if you want to get Hi, well, you can obtain it from here. You assume that the sigma, the magnetizations are the ones of the data. And so here you know the J, you know the magnetizations from the data, and you can solve these for the fields. Okay, so this is a very simple procedure. You don't have to do any Monte Carlo. You just invert. You get from the data the correlations and the magnetizations. You invert the correlation matrix. This gives you the coupling. And then from here, you get the fields. So this is very efficient computationally. The problem of this, the disadvantage, is that if you then, it depends on what you want to do. So I will discuss it a bit more systematically later, but once you have this model, you can do two kinds of things, mostly. One is to try to use the parameters to infer information. So for example, from the J, you get the direct coupling between two variables, which is a good measure of their interaction. So if you want to do that, the mean field is usually a good approximation. It gives you a good approximation for the couplings. But the other thing you might want to do once you have this is to generate new configurations of your system. So you might want to use this as a generative model, which means I learn a probability distribution from the data, And then I use my probability distribution to generate new data. For example, if you have proteins, maybe you want to generate artificial proteins. In that case, mean field is usually not a good thing, in the sense that when you go, if you take the mean field parameters and you put them here and you sample new configurations of the sigma from the distribution, what you see is that the new configurations that you are sampling are not very similar statistically to the training data. So typically the moment match, for example, if you look to the moments, they are, even if you infer by matching the moments, because you are doing an approximation, the moments will be quite different, and if you look at 3-point correlations, they will be also different, and so on and so forth. So mean field is good to get an idea of the parameters, but it's not particularly good if you want to use it as a generative tool. Okay, we will come back to this. So, yes?
So you mean when we use mean field approximation, we don't need to do the Boltzmann learning and these iterations?
Mm-hmm. Exactly.
Actually, we can find the solution of the problem.
In one shot, basically. I give you the data, I will show you an example. I give you the data, you compute from the data these two quantities, which is easy, you just do an average over the data of these things. And then from this, the Cij is just, Cij is sigma I, sigma j minus sigma I, sigma j. So if these are given by the data, you have your estimate of Cij from the data. And then you have an L by L matrix that you have to invert. So inversion of an L by L matrix takes L cube or something like this. So you get this, and then these are L equations that you solve. So it takes time. That is something like L cube. Here, the problem is that the Monte Carlo can take a time much longer than L cube. So that's the issue.
But the point is that mean field approximation is not a good approximation always, right?
Exactly. So
and we need to do Boltzmann learning.
Exactly. So it depends on what you want to do. Mean field approximation is, in many applications, it's a good approximation to get an order of magnitude of the couplings. So if you are just interested in knowing, for example, which are the strongest couplings, then it's a good idea. It's a good approximation. But if you make mistakes on the couplings, so if you put the couplings back here and you sample new configurations, the mistakes you make will accumulate, and the configurations that you sample will be quite different from the training ones. So if you want to generate, if you want a good generative model, what you want is that you learn a probability distribution that is able, by sampling, to generate states that are similar to, for example, if you do image, you start from images, you learn a distribution, and then you use the distribution to generate new images, and you want the images that you generate to be similar to the ones. So if you do mean field, this is typically not the case, okay? It's not a good generative model. Okay, so what else can we do? We can do pseudo-likelihood maximization. So I don't have enough blackboard, so let me erase this. So how does it work in this case? It's very easy. So you start from the pseudo-likelihood. So what you want to learn when you do pseudo-likelihood maximization is you directly want to learn from the data the conditional probabilities, okay? So what you do, as we did for Gaussian variables, you define a pseudo-likelihood, which is 1 over m times the sum over your data of the logarithm of the probability. Sorry, this is, well, okay, this is of the probability of sigma I conditioned to the other variables. So here you have data. So on each data point, you have the value of sigma I, and you have the value of all the others. So you compute this, and you maximize the pseudo-likelihood. So you will have one pseudo-likelihood per variable, and you can do it separately or you can sum them. It's the same. Why? Because this thing is 1 over m. So when I take the logarithm of this, I get hi sigma I minus, so, and this will be minus the log of 1 plus this. But you see that hi, the local field acting on variable I, depends only on the field associated to the same variable and the couplings associated to that variable. So this quantity does not depend on all the fields and the couplings. This depends only on hi and the vector ji that contains the couplings of I with the others. So this is one row of the matrix Jij. So this means each pseudo-likelihood associated to each variable, you have L variables. So you can imagine that you have your string of L variables, sigma 1, sigma 2, sigma L, and you want to learn the probability of sigma I condition to the others. So you will have a local field Hi, and you will have couplings Jij. And from each pseudo-likelihood, you learn one field and L minus 1 couplings. And when you put them all together, you will learn the whole thing. But because each pseudo-likelihood only depends on these two things, it's the same to maximize each pseudo-likelihood separately or take the sum. You could take the sum over I and maximize all together. It's exactly the same. Okay? Now, what is the advantage here? The advantage is you see that you don't have the partition function here. Why? Because, well, here, in order to normalize the joint probability of all the variables, you have the partition function, and the partition function is the sum over all the variables of the numerator. So this is a sum over 2 to the L states, so it takes exponential time to compute, and so you have to sample from Monte Carlo. But here, if you fix L minus 1 variables, when you normalize, you only have 2 states for the variable sigma I. So the sum over 2 states can be done, and you get 1 plus this. And so everything is explicit, and you don't have to take this normalization. So this means that the pseudo-likelihood can be computed explicitly, and this is the expression. So now when you take derivatives, you can write explicitly the derivatives. Okay, maybe I will not do it, but you have it in the book. So when you take the derivative with respect to hi, for example, here you have an hi, so you will get something like 1 over m sum over m of sigma im. And here you have minus 1 over m sum over m. Here you have 1 over 1 plus a. When you take the derivative, you have something like this. And this should be 0. So you see that the derivative of the pseudo-likelihood, well, okay, I'm doing it, so let's do it. Let's do it just for the fields. The derivative of the pseudo-likelihood gives you a sort of a moment matching condition, which actually corresponds to the Cullen identity. So this is telling you that the expectation over the data of sigma I should be equal to the expectation over the data of, this is sigma im times the conditional probability of sigma im. Okay? So this is the same as the Cullen identity I discussed yesterday, but with the average over the model replaced by the average over the data. Anyways, this is not very important. What is important is that you can compute this gradient from the data. So you can do gradient ascent on the likelihood. So at each step, you will do something like dhi over dt equal to some learning rate, let's say 1 over the learning rate times the hi over dt is equal to the gradient, so you do gradient ascent, and at each step you have couplings, you have h and j, so you can compute from the data this hi. The problem, you see that this is very good if you don't have too many data, because if you have a lot of data, then here at each iteration you don't have to do the Monte Carlo sampling, because you can compute everything exactly from the data, and you don't have noise. This is a deterministic equation once you have the data, you see. So you don't have to worry about sampling Monte Carlo and sampling noise and so on and so forth. But if you have many data, at each iteration of the algorithms, you have to compute these things from the data, and so it can take time. But this can be solved. You can do mini batches on the data and things like that. So in general, this will converge quite fast, okay? Yes?
How does the sigma appear in the second?
Yes, because hi, because there is this, well, no, you're right, there is no sigma here. This is wrong, sorry. You're right, thanks. There is no sigma. So yeah, it should correspond to the Callen identities, so let's check, but yeah, it's correct, it should be like this. Thanks. Yeah, I'm differentiating with respect to hi, so there is no sigma. No, it's correct. So this is the correct equation, sorry. And the Callen identity has no sigma, so it's okay. Thank you. Okay, so the advantage of pseudo, so let's erase this and also this. So the advantage of pseudo likelihood is that it's computationally more efficient than Boltzmann learning. But again, the disadvantage is that, and okay, you get good estimates of the couplings and the fields, much better than in field. The disadvantage is that, again, it's not easy to use it to generate new configurations. Why? Because, okay, first of all, the problem is, you remember, you can show that if you have infinite data, then pseudo-likelihood maximization is the same as Boltzmann learning. And this is, we showed it for Gaussian variables, but it's true also for, in this case. So if you have infinite data, this maximizing the pseudo-likelihood will give you the same as Boltzmann learning, but okay, so in that case it's okay. But if M is finite, then the problem is that your matrix Jij has to be symmetric. Here you are inferring each row is being inferred by different data. And so in general, you will get a non-symmetric J because of noise, of the finite sampling that you have. So first of all, you don't know how to put it here, because here the J are symmetric by construction if you have different, because you see J is multiplying sigma I, sigma J. So even if it's not symmetric, because this is symmetric, the anti-symmetric part will disappear. So then you have to decide what to do. One thing you can do is to symmetrize by hand. So you can say, okay, for example, I put in the distribution the average of Jij and Jji. And then I plug it here and I try to generate new data. So people tried, and it's not very good. So once again, because of the small mistakes you make on the J, when you use it as a generative model, you don't have a very good correspondence between what you generate and what was in the training set. The other thing that you can try is to say, I keep it as it is. So I forget about the joint probability. I know the conditional probabilities. So you can try to generate new data by saying, okay, I start from a sequence. I choose, so I have sigma 1, sigma 2, sigma L. So I start from a random sequence. I choose a site, I, and I say, okay, now I know the probability I learned the probability of sigma I condition to the rest. So I draw a new sigma I from that probability, okay? So now I choose another site, and I draw this one from the conditional probability. So I use the set of conditional probabilities to sample new states, like I would do in Gibbs sampling. I do it many times, and I hope that I converge to something good. Well, we tried that, and it doesn't work. So typically, the problem is that your conditional probabilities, because they are learned from finite data, are not exact. So you have mistakes, and you have inconsistencies between them, because the couplings are not symmetric. So they don't respect the fact that they should all come from the same joint distribution. So you make mistakes when you do this sampling, and that accumulate, and then your sampling goes to states that are not statistically similar to those that you have in the training set. So once again, I'm not going to, I mean, if you're interested, I can show you, offline, I can show you results, but I'm not going to discuss this too much. But then, so once again, this is not a very good model for to generate things. And this is something that is also, I think, used in more like modern machine learning architectures, like language models. So you see that pseudo-likelihood maximization is very similar to what you do. I mean, for those who know language, I think you all know language models. So it's very similar to what is called masked language modeling. So you have your string of text, and what you do is to learn a model that will learn to predict a word based on the context of what is before and what is after. So again, you try to learn the conditional probability of a word given the rest of the string, and you train your model by randomly masking some sites and learning these conditional probabilities. So pseudo-likelihood is very similar. So typically in these like modern architectures, you have a string of text that is input in a deep network of like transformers and stuff. And so you start with your text that is here. You input this in layers of stuff. Okay, and then you input this, you do many things, and you end up with logits at the end that give you the probability, that are trained in such a way to give you the probability of one word in the context of the rest, right? So this is what pseudo-likelihood does. Here it's a very simple architecture where you have your data, right? You can see that you can represent this as you have your data, sigma 1, sigma L. You apply a very simple linear layer, because what you do here, you produce the fields by doing a linear combination of the input plus a constant. So this is what a linear layer would do. So here you have, if you want, a linear layer that will take these as inputs. It will produce the HI here. So H1, HL. And what you do is to train this. These HI are the logits that you train to reproduce what you have in the output. So you take the same configuration here, sigma 1, sigma L, and you learn this in such a way that H1 will give you the correct probability of sigma 1, right? So it's a very similar idea, except that this is just one linear layer plus a softmax, if you want. Here you do a softmax. So pseudo-likelihood is like masked language modeling, where you have a linear layer and a softmax, and you have also here you work with fixed size, while in language model you try to work with strings that can have a variable size, so you have all the problem of positional encoding and so on and so forth, but the idea is similar, okay? So also in language modeling, what you know is that if you try to generate a text by taking a starting text and predicting the words at random based on what is before and what is after, it doesn't really work. what people do when they want to generate new text is to use a different procedure, which is called autoregressive generation. So how does it work? It's basically a variant of this, in which you say, okay, instead of using the conditional probabilities of each variable in the background of all the other variables, I change my setting. So autoregressive models. So in autoregressive models, what you do is you use the fact that the joint probability of L variables is always can always be decomposed as a chain of joint of conditional probabilities. So you say, okay, my P of sigma is not this one now, so forget about this one. I will write it as P of sigma 1, P of sigma 2 condition to sigma 1, P of sigma 3 condition to sigma 2, sigma 1, up to P of sigma L condition to sigma L minus 1, sigma 1. Okay? This is exact. You can always do it. And now you say, okay, let's try to learn these things. Okay? So I do maximum likelihood. So my likelihood is going to be 1 over m sum of the log of P of sigma m, where sigma m are my data. And, well, because this is a product, you get a sum of likelihoods. Let's say L1 plus L2 plus L3 plus LL, where each likelihood is the average of the log of one of these terms. And now what you do is to make a model for this and try to learn each term, okay? So the simplest model is always the same. If you have this kind of variables, you can say, okay, the probability of sigma I conditioned to what is before. Now, it's not conditioned to everything, but only to what is before. So now you are changing the architecture. The probability of sigma I conditioned to what is before can be written as exponential of a field times sigma I divided 1 plus exponential of the field. This is always the case for binary variables. And then I write the field in the same way. So I write this field as a local field hi plus the sum over j, but now smaller than I, not different. Before it was different from I, of jij sigma j. Okay? And then for each term, I have a field. So I have h1, h1 is just a field. h2 will be h2, capital H2 will be a field h2 plus j 2, 1, sigma 1, and so on and so forth. So what you're saying is that, again, this is an autoregressive architecture where each field is a linear, each field, capital H, is a linear combination of the input, but the input is restricted only to the previous ones. So for example, H2 will depend only on sigma 1. So here you get an autoregressive linear layer where H2 depends only on sigma 1, and H3 depends only on sigma 1 and sigma 2. Sorry. So let's say H1 is a constant, H2 depends on sigma 1, H3 depends on sigma 1 and sigma 2, and so on and so forth. And the last one will depend on all the previous ones. Okay? When you train this by maximum likelihood, when you maximize the likelihood, you get equations that are very similar to the ones of pseudo likelihood, with the only difference that these sums are only on the previous ones. And you see that each of these likelihoods in this architecture depend on different parameters. So the first one depends on small h1. L2 depends on small h2 and J21. L3 will depend on h3 and J31 and J32 and so on. So each term can be maximized separately. So basically you are learning separately a model that gives you the probability of a spin conditioned to what is before. The number of parameters, however, is the same. I mean, the thing is, if you do Boltzmann learning, you have, okay, you always have L fields, and then how many couplings you have. If you do Boltzmann learning, the couplings are symmetric, so you have L times L minus 1 over 2, and also mean field. If you do pseudo-likelihood, the couplings are not symmetric, so you have L times L minus 1, so you have twice the number of couplings, but this is not really consistent, so you should symmetrize them or should do something. And if you do autoregressive, you still have L times L minus 1 over 2. Basically, your matrix is lower triangular instead of symmetric. You say that there is 0 couplings with the future and there is a coupling with the past. Okay? The good advantage of the autoregressive models is that they are generative, because once you have these parameters, you have a consistent way of generating a new configuration, which is just you generate the first spin, then you generate the second spin conditioned to the first, and so on and so forth. So now maybe what I can do, is it clear, the architecture? I can try to do some table, I don't know if you want to compare the different methods. So let's say you have Boltzmann learning, you have mean field, you have pseudo-likelihood maximization, and you have autoregressive models. And, well, you can ask a few things. I mean, you can ask many things, but for example, you can ask the computational efficiency, and well, mean field is very fast. You just have to invert the matrix. These two have similar computational efficiency, and it depends a bit on how many data you have. If you have many data, it can be a bit slow, but then you can use batches. So this is good, let's say, a bit slower than mean field. And Boltzmann learning instead is quite slower than the other methods, okay? Still doable if you have a GPU, and if you have, I mean, also on a GPU actually, but it takes a few days. And you, I mean, it's a bit slower. Then you can ask what kind of information you can get from the couplings and the fields. And this is, I would say, comparable for these three methods. So if you, for example, try to look at the biggest couplings that you have, we will see in proteins that the biggest couplings are related to contacts. You have good prediction of contacts from all of these methods. Pseudo-likelihood maximization is a bit better than mean field. You can discuss, but they are all good. From the autoregressive, it's a bit more complicated to interpret the couplings, because in these three schemes, you are really inferring a graphical model, even if this is exact and this is approximate. Okay. In the autoregressive case, it's a bit different in the sense that you're not inferring this model. So it's not, the Jij are not really the couplings. You are inferring this chain of joint probabilities. And so if you want to get the coupling strength of two variables, it's a bit complicated, because what you should do is, for example, you should take the log of the probability, call it energy somehow, or Hamiltonian. But this will be a sum of all these terms, and you can see that effectively you have many body interactions, because you have, I mean, even if each of these terms is written as the exponential of two body interactions, in reality, in the denominator, there is the field that depends on all the others, and you can see that this will induce many body interactions. Maybe I will not go into the details, but so if you want to get couplings, what you have to do is to start from a sigma, from one state, and change two spins, and see what is the variation of energy when you change two spins. And so you can extract a coupling from there, but the couplings will depend on the rest of the spins, because you have all this. So you can get effective couplings, and they carry similar information if you do that. But okay, you have to be a bit careful. So let's say you have to be careful, okay? This is not a graphical model in the same sense as this. So you have to decide then what you do with your model.
So you're saying that we have to fix or take the couplings pair to pair, fixing the others, and see what is the effect on the energy?
Yes, this is one way of doing it. So you take a reference state, you change two spins. For example, you start from all 0, you switch I and j to 1. The difference in energy should be, if you want to interpret this as a Hamiltonian of this form, when you switch on two spins, the difference is, the energy will be jij plus hi plus hj. So then you remove hi, hj, and you get jij. So you can infer a coupling. The problem is that the coupling that you get depends on the state of all the other spins, because you can see that these denominators that you have here contain effectively many body interactions. So you have to be careful. Yes?
In that, we will have the same performance or similar performance if we take like random states, initial states.
You mean to estimate the coupling?
Yes.
Yeah, I don't know. It really depends on the problem you have. That's why I'm saying. Okay. We tried on proteins, and on proteins it works in the sense that the couplings don't depend too much on the initial state. So you can take one reference, you can average over some initial states. But then if you have another problem, it could be, so I'm just saying, be careful that the hi and jij that you get are not one of the graphical model. They are different, so you have to decide what to do then. And sorry, maybe here we can say this is training computational efficiency. Then the other thing is that now you can, once you have these models, you can say, let's try to generate configurations. So if I generate new states, these ones will work. These are consistent probabilities that will give you good states. So for example, if you generate many states and you look at averages of observables, they will be the same on the generated states and on the training set. These typically do not work, okay? They are not good at generating new states. And on top of that, you can ask, what is the computational cost to generate a new state? And here it's slow, because if you want to generate a new state from the Boltzmann distribution, you have to do it by Monte Carlo, so it can be slow. So the good thing of autoregressive models is that it's very fast, because you can generate in this sequential way. So you start from a sigma 1, then you go on. And at each step, in order to compute the conditional probability, you have to compute this local field that takes order L operations. So in the worst case, it takes order L square operations to generate a new state. While in the Monte Carlo, it could be even exponential in L if you have, if your system has metastable states and so on. And well, for these two, I don't say it because it's the same as Boltzmann learning, but it's not even good, so you don't want to do that. So, okay, so depending on what you want to do, you can choose the best approach. there is one last thing, which is, and I will show you an example of that as well, which is the dynamics. So you might be interested not in generating states in equilibrium from this probability distribution. You might be interested, and I will show you why, in starting from a state and generating a dynamical process that starts from that state. For example, by proposing mutations and accepting them with this probability or this probability. So in that case, it's much faster to do it with Boltzmann machines than with autoregressive model, because autoregressive models are very good if you want to generate a new configuration from scratch. Because you say, okay, I don't have a configuration, so I generate sigma 1, then sigma 2, then sigma 3, then sigma L, and it's very fast. But if you want to propose a mutation and accept it with this probability, then it's very complicated, because if you have a state and you want to change one sigma in the middle, you have to recompute all the probabilities of the following spins. So it takes a lot of time. Okay, so if you want to do one mutation of your configuration with this, it's very slow. While with this, you know that if you want to do Monte Carlo and you do a single spin flip, you just have to compute the energy change and you do Metropolis, and it's very fast. So really, depending on what you want to do, each of these architecture has advantages and disadvantages. So these are complementary approaches, and you can choose your favorite one. Okay? So there is one last thing I would like to say. Yes?
Can you explain again, please, the difference between the last two ones, computational cost generation and dynamics?
Yes. So you can say, so when you want to generate a new configuration, you would like to generate one independent sample from P. Okay? So you don't have any previous information. You say you have a probability, you want to sample one configuration from this probability. Here, it's very easy, because I draw sigma 1. I have two states. I know the probability. I draw 1. So I know that my first spin is 1, for example. Then I know the probability of sigma 2 conditioned to the first. So I draw sigma 2, and again, you have to do one operation. You have to compute the field, and then you have to do one operation. Computing the field, I erased it, but the field is a linear combination of, so it's of order L. So at each step, it takes of order L operations to compute the field, and one operation to draw the new state. So you have L states, each one takes L steps, so you have L square operations, and you get a new configuration. Here instead, you don't know how to sample from this distribution, because the partition function is, because they are all correlated, no? The spins are all correlated. So you don't know how to start with 1, because you should compute the marginal, but to compute the marginal, you have to sum over 2 to the L states. So what you do is you do Monte Carlo. You start with a random state, you propose a flip, you accept with the Metropolis step, and you do it many times until it converges to equilibrium. The problem is that it could take exponential time to converge to equilibrium in L if your system is disordered and metastable and so on. Okay, this is okay. If you have experience with Monte Carlo, this will talk to you. Otherwise, not really. But you have to try and see. So it will take a lot of time. So this is where I'm saying generating from scratch one equilibrium configuration is slow for Boltzmann learning and is fast for autoregressive models. I mean, can be slow, then it depends on the problem. But instead, if you already have a state and you want to know and you want to propose mutations to that state, for example, I will show you in proteins, this can be a way to model evolution. So I start from a protein I know, and I want to propose mutations. So then I want to know, I have a state, I change one spin. How does the probability change? Okay? Here it's easy, because the partition function is fixed, is a constant. So if I want to know the variation of the probability, this depends only on the numerator. And if I change one spin, I have to change L terms here. So it takes order L time to compute the change in probability. And then I can accept or not with metropolis weight. Here it's lower because if you change one spin, for example, if you change sigma 2, this will affect this term, but it will also affect all the following terms. Okay, so you have to compute to compute the variation of p when you do one mutation. You have to compute the variation of all these. It's not much. I mean, it's a factor L slower basically than Monte Carlo. But so if L is 100, you lose a factor 100 in the time it takes to propose a mutation. Sorry, not to propose, to check the effect of a mutation. Okay?
So in the dynamics, we want to check the energy variation between two.
Exactly. So the energy is the logarithm of the probability. So in order to check the variation of energy, you have to check the variation of the probability. Here it's very easy because the log of the probability is just this plus a constant that will not change.
The first case is just in one step, right?
Well, L steps, because if you change one spin, you have to compute the effect of the change of all the other. Each spin is coupled in principle to L spins. So it takes L operations in Boltzmann distributions. Here it takes L square operations because you have L terms and each of them is a sum of L things. So, okay. Anyways, this is just to give you an idea. Now, the last thing I want to discuss, and then we go to the protein application, is regularization. And maybe let's just do very quickly. So you can do. Up to this moment, I didn't put any regularization. So in principle, I am inferring a model with L square couplings. I might want to, maybe I might suspect that my coupling matrix is sparse. For example, if you want to infer, I don't know, a two-dimensional Ising model, then your couplings are non-zero only if you have nearest neighbors. So many couplings are actually 0. So you might want to enforce that. So there are many ways. You can add an L1 regularization to the likelihood. So when you do, for example, in Boltzmann learning, when you do your gradient ascent, you can add an L1 norm, and you can regularize that way. There is also something we did over the past years that I want to just very quickly explain that works quite well, which is to activate the couplings one by one or start from a fully connected model and remove the couplings one by one. So how do you do that? So for example, if you activate the couplings, the idea is very simple. So you start from a model with 0 couplings. So you start with j equal to 0. And if you start with j equal to 0, you only have the fields, and you can learn the fields very quickly by, as I discussed yesterday. So if you don't have couplings, the fields are just the logarithm of the frequencies normalized. So you have the fields at the beginning. And then you can ask, okay, if I add a coupling, what is the variation of the likelihood? Okay? So if I say my Hamiltonian here is equal to minus sum over I of hi sigma I minus sum over ij of j sigma I sigma j, and in such a way that this is e to the minus h over z. Okay, the minus sign is the convention of physics. Now, okay, so let's say I start from some h0. For example, h0 might be the one without any coupling, and I add the coupling. So I say h is h0 minus, I add one coupling, ij. Okay? And the question is, how does the likelihood change? So the likelihood, the variation of the likelihood is 1 over m, sum over m of the logarithm of p minus the logarithm of P0, right? Where P is the exponential of minus h over z, and P0 is the exponential of minus h0 over z0. So what is this? This is 1 minus m times the sum over m of minus h of sigma m plus h0 of sigma m minus log z plus log z0. Okay? Now, the difference h0 minus h is simply jij sigma I sigma j. Right? So the first term is jij times the average over the data of sigma I sigma j, which is something I have. And this term is plus, well, we can write it as minus the logarithm of z over z0, where z and z0 are constants, so when you do the average over the data, they remain constants. Okay, this seems difficult to compute because it's a ratio of partition functions, but actually, if you look at what it is, z over z0, you can write it as the sum over the sigma of e to the minus h divided by sum over sigma of e to the minus h0. But h is h0 plus jij sigma I sigma j. So this can be written, this is the partition function of h0, and this is, if you factorize the exponential, this is the average over the average of jij sigma I sigma j over the model h0. Okay? So now the average of this is actually very easy to compute because I will erase this, okay, to make some space. Because jij is either 0 or 1. So this is e to the jij. If sigma I sigma j is equal to 1, this is e to the jij times the probability that sigma I sigma j is equal to 1, which is basically the expectation of sigma I sigma j over h0, plus if sigma I sigma j is equal to 0, this is 1, and the probability that sigma I sigma j is equal to 0 is 1 minus this. Okay? So this thing in the end is given by jij fij, where fij is the frequency of sigma I sigma j being both 1 in the data, minus the log of e to the jij P0 ij, where P0 ij is the probability in the model of sigma I sigma j being both active, plus 1 minus P0. So if you are doing Boltzmann learning, you know during the Boltzmann learning, you have to compute, you know the frequencies of the data, this you compute at the beginning, but you also know the probabilities of your model because you are doing Boltzmann learning. So the idea is you start without couplings, you do Boltzmann learning, and from time to time, you look at your couplings, and for each pair ij, you compute the variation of likelihood that you would have if you activate that coupling. So you have it, and so you can maximize this over jij to find the best possible value of jij, and you can compare all the possible ij, and you can choose to activate the couplings that maximize the likelihood. So you can say, okay, at the beginning I have no couplings. I look at this, I decide, I don't know, 10 couplings that are the best ones. I switch them on, and then I do my Boltzmann learning only with those couplings. The other are kept fixed to 0. I converge, and then I look again, and I activate 10 more, and so on and so forth, okay? Until you like the result. What does it mean you like the result? You can, what we did, I mean, this was our choice, but you can find a better choice if you want, but what we did was you can do two things. One is to check usually we check the Pearson correlation between the correlations of the model and the correlations of the data. So you have L square over 2 correlations. You can do a Pearson correlation of this. So you do this. And so usually we check this. And when it reaches something like 95%, we consider the model to be good. This is just based on our experience. And what you see, essentially as a function of the number of couplings that you have, let's say, if you look as a function of the, how can I call it now? Well, okay, number of couplings, fraction of couplings that you have activated. This usually does, if you plot this quantity as a function of the fraction of couplings, this means this is without couplings, and this is with all the couplings present. This usually you get something like this. Okay? And this is about 95%. I mean, 95% is what you typically get from the best model because you always have a bit of noise. So you can say, okay, maybe I stop when I reach 95%. But there is something else you can do, which is to look at the entropy of your model as a function of the fraction of couplings. And what we see is that actually the entropy has a maximum at some point, and then it goes down. Why? Because if you have too few couplings, in order to describe your data, you have to make them very strong. And so in the end, you lose entropy. And well, if you have too many couplings, you are overfitting the noise, and you are again reducing the entropy. So one possibility to select the best model is to look to a model that is sparse, and that's the fraction of couplings that gives you the maximum of the entropy. And now I think this starts to decline a bit after the peak. So the peak is in the region where you already saturated to the good performance. So by looking at the entropy, you can find a model that is fitting well the data and maximizing the entropy, and so you can hope that you are not overfitting the noise. Okay? So I think this is at least for this kind of models, this is a good way to regularize that you can do online during the Boltzmann learning. So we have a package that does this, that is public, and you can play with it. So you can do it either starting from 0 and activating the couplings one by one until you reach the peak, or you can do it starting from the model with all the couplings. You do the Boltzmann learning, you converge, and then you start removing the couplings one by one, and it's very similar. You can check which couplings are the ones that are the most irrelevant by a similar calculation, and you remove those that are irrelevant until you see that the entropy goes up, and then you see that at some point the performance will go down, and then you can decide where you stop. Okay? Yes?
What is the role of the variation of the likelihood here as to accept one coupling? Or
when you activate or when you decimate?
The difference of the log likelihood that was computed there?
Yes. We use it to select which couplings to activate. We activate the couplings that maximize the variation of the likelihood.
Okay, so we just that coupling if the variation
Well,
is
you could do it in that way, but we do it in one shot. So we do the Boltzmann learning, we saturate with a given choice of couplings, and then we say, okay, we look at all the couplings that are 0, we compute this for all of them, and we sort them, and we take the, I don't know, the top 10 or 100, it depends on, this is a parameter in your learning. So you take the top n couplings, and you switch them on. And then you keep doing Boltzmann learning for a while until you converge, and you do it again. And the point is that estimating the change of likelihood is very easy because from the during the Boltzmann learning, you already have all the ingredients you need, so you can do it without cost, basically. Okay.
Oh, excuse me, my question. What did you do here in that you're getting here?
I'm just, this is true only for binary variables, but you can generalize to multi-state. But if they are binary, sigma I, sigma j is either 0 or 1. It is 0, it is 1 with probability given by this. So with probability, because this is the expectation value of sigma I, sigma j is either 1 or 0. So the expectation value of a binary variable is the probability of the variable being 1. So with probability given by this is 1, and so this is exponential of j. And with probability 1 minus this is 0, and so this is 1. That's it. And if they are multi-state, you can generalize this thing.
What was rho again?
Rho is the Pearson, okay, yes, sorry, I was fast on this. So when you train these models, then what you can do is you take the Cij of the data and the Cij of the model, and you do a scatter plot, and typically you have something like this with points. So you have L square points for each pair. You can compute the correlation of the data and the correlation of the model, and you have a cloud of points, and you can do a Pearson correlation of these to understand how correlated they are. So 1 would be perfect correlation, 0 is no correlation. So if you have 0, it means your data are like this. And if you have 1, it's a perfect correlation.
So rho is between 0 and 1.
Yes, it's a number actually between -1 and 1. It's a Pearson correlation. So if they are like this, it can be -1, but hopefully it's not the case. So you typically start from 0 and you go up to something close to 1 when you train the model.
And one last question.
Sure, yeah.
Here, you're just turning on to one coupling, or you're summing over I and j?
No, it's one coupling.
It's just one coupling.
So for each coupling, you can ask what is the change in likelihood if I turn on that one, and then you can take the top n.
So you're not summing.
No. Okay. Now I can introduce. Okay, I think I'm done with the theory, and now I can introduce the application of these ideas to proteins. I mean, of course, you can apply these ideas to many data, and in particular, I told you the data of the neural recording of animals can be used in this way. This was done by many people, but in particular, first by the group of Bill Bialek, then more recently by Rémi Monasson and Simona Cocco, and then many others. Ulisse Ferrari, well, I mean, I can make a list of people working on this from the neural point of view. But I will discuss now the application to protein sequences, and also it can be done also with DNA or RNA sequences. So let's see. Okay, so now I will use slides because I want to show you figures. So these slides were prepared for a summer school last year, and oh, what's going on? Ah, no, this is not what I want. Okay. Okay, so okay, now I want to explain how you use these ideas in biology, so in particular in protein, to analyze protein sequences. So what is the goal? So as you know, I mean, proteins are chains of amino acids, polymers made of monomers that are the amino acids. So you can represent a protein by a string of letters, and the letters represent the 20 possible amino acids. This, in principle, this encodes all the properties of your protein. So the proteins are produced as a linear chain that will then fold into some complex three-dimensional structure, and this is due to the interactions between the amino acids. So they will fold. They first produce what is called secondary structure of helices and loops and so on, and then this structure will fold into a 3D structure. Once the protein is produced and folds, then it will perform some function. For example, you can imagine, we will see some proteins that are used by bacteria to resist to antibiotics. So the protein will be transported to the membrane, and then when the antibiotics arrive, the protein will bind to them and cut them and degrade them. Okay? So this is what is called the function. So the idea in biology is that you have a mapping from sequence to structure to function. And what you would really like to know is, given a sequence, what is the function of the associated protein. The problem is that, well, the mapping from sequence to structure is somehow a mathematically well-defined problem, because you start from a string of letters and you want to map that into a physical protein, which is a collection of coordinates of atoms. So both things are mathematically well-defined, and you have many data. So we have now, I mean, this keeps growing, but we have of the order of 10 to the 8 protein sequences that are available in databases, and we have of the order of 10 to the 5 structures. So the problem of mapping sequence to structure is quite well defined, both from the point of view of mathematics and of data science, if you want. And I mean, thanks to that, it was solved, as you know, by essentially solved by AlphaFold. I mean, it's not, there are still many problems. I mean, in some cases it doesn't work, but anyway, now you can have reliable predictions of structure based on the sequence using these deep architectures. And of course, there was a Nobel Prize for it. So the problem is that the mapping from structure to function is much more tricky for several reasons. First of all, because function is not really a mathematically well-defined thing, because it depends on many variables. It depends on the environment. So if you have a protein and you put it in a certain bacteria, it depends if the bacteria can express it, can transport it to the membrane. Then it depends on the chemical conditions, whether it binds or not. So it depends on a lot of variables that can affect the result in a pretty uncontrolled way. So I mean, when you talk about function, it's really hard to define it properly, what you mean. But even in the simplest case, so you can say, okay, now I do a well-controlled experiment. I have one species of bacteria. I put my protein in there. I have controlled chemical conditions. I put a certain antibiotics in a given dose, and so on and so forth. So everything is well-defined. Still, measuring function is hard. So you have to do experiments where you grow this bacteria in very controlled conditions. You put antibiotics, you select, you have to define a growth rate, and so on and so forth. At the end, you come up with a number. So for a given protein, you can have a number. For example, the growth rate of a certain bacteria in certain conditions, and that's a number, okay? So you can get a number, but getting this number is very expensive. So typically, these are costly experiments, both in terms of time it takes and money it takes. So we don't have many data. We have experiments where people are measuring functions of some proteins in some conditions, and there are more and more such experiments. But still, we are in a very, very, compared to the structures that are available, we are in a very scarce data regime. Okay, we have few data, and there will be more and more data, but not so many data. And again, the nature of the data is subject to interpretation, because you have to, when you compare different experiments, you have many things that are different, and it's not like structured. I mean, structure is structure, and kind of. I mean, there are proteins that can have multiple structures, but it's still a very much better defined thing. So this is to say that the mapping of sequence to function is still a very open and unsolved problem. And, well, you can either decide to do it in steps. So you can say, OK, I solved the structure, and then I can try to map structure to function. And there are people who work on that. But what we are doing is to try to kind of skip structure and go directly from sequence to function by using these kind of models I discussed before. Why? Because you can include structure, but it's still quite computationally expensive to include structure in the game. I will maybe discuss later why. And so, I mean, we try to do things in the simplest possible way. And given that we have few data, and we try to start from the simplest thing. So what I will try to do is to show you ways to predict function starting from sequence using these simple models and the few data we have. So why we want to do that? Well, because if you learn something about the sequence to function relationship, then you can do many things. You can, for example, design artificial proteins that have certain functions you might want to have. You can try to understand evolution, so try to understand how nature evolved certain functions. And this means you have to understand also the impact of fluctuating environment, phylogeny, and all the processes that go on in natural evolution. You might want to make models to understand what happens in the lab when you do an evolution experiment in the lab. And these are more and more being done. So you try to take a protein and you try to evolve it to develop a certain function, for example, or to differentiate and explore the space of sequences compatible with that function. So we try to do models to do that. And then you would maybe want to optimize protocols. For example, if you want to design a library of sequences that have certain properties, is it better to do it with constant selection, or maybe alternating cycles of low selection and strong selections, things like that? And the other thing is that we would like, and people are doing it, to generate mutational paths that connect different proteins and try to see, for example, how they can switch from one function to the other, how easy it is to switch, and things like that. Okay, so we try to, I mean, together I work on this together with Martin White mostly, and many other collaborators. We have a web page, but there are many people, of course, that are working on this kind of things. So it's, when I mean we, I mean the community, of course. Well, this is already done, the advertisement of the book. You have the book. So, okay, what are the kind of, maybe I will discuss the kind of data we can use, and then we stop, and tomorrow we will continue the discussion. So the kind of data we have are natural data. So you have these databases where you can download proteins that have been sequenced. People sequence proteins for all kinds of reasons. So typically they put them in these databases, and you can get the sequences. And what you, I mean, these databases are organized in families. So you typically have families of sequences that have typically the same structure and similar function, okay? So for example, this is this beta-lactamase protein that is used by bacteria to resist antibiotics. So you can get an alignment of sequences that are all folding into the same structure, and they are doing something similar, so resisting to antibiotics. If you look at this, so you can align these sequences. I will not enter into the problem of sequence alignment, which is a domain in itself, but you can align them with standard methods. So when you align them, you have to introduce sometimes gaps, because during evolution, maybe one amino acid can be removed. So you can have a gap here, because here there is one more symbol. And so you have 21 now symbols in your strings that are the 20 amino acids plus the gap. And if you typically look at one family, you have something between 100 and 10 to the 5 sequences, so between 100 and 100,000. And the striking thing is that they are very different. So if you if you take two sequences at random from their alignment, they typically differ by 70–80%. Okay, so if you look at the number of mutations, how many amino acids are different between the two, most of the sequences is different. So this means that there is a lot of variability. So nature found many ways of generating the same function with very different protein sequences. So the idea is to use this as a sample. So this will be our data. So the length of the protein is L, typically about 100, and we have M sequences that will be typically between 10 to the 2 and 10 to the 5. And the idea is to use this as our data to train a model, and we will train the model by maximum likelihood. So the idea will be that the model will give high probability to the natural sequences with the idea in mind that these are sequences that work. So we want to give high probability to the things that work. In statistical physics language, energy is minus log probability, so high probability means low energy. So the idea is we try to learn our model in such a way that the natural sequences will have low energy. And then we have other kind of data. So there are experiments where people take one protein and they do all possible single mutations of that protein. So on each site, now you have 100 sites, for example. On each site, you put all the 19 amino acids that are not in your reference. So you have 19 times 100 measurements of single mutations, okay? And you can measure in these experiments, these are called deep mutational scans. You can measure in these experiments the fitness, where fitness again is a measure of function, and again it's not very well defined, so you have to interpret the data carefully, but it's typically the growth rate of some bacteria that have this sequence inside in some conditions. So you have a number for each sequence, and this is supposed to give you an idea of the impact of one point mutation on a certain sequence. So from the point of view of the space of sequences, what you're doing here is taking one natural sequence, doing all possible mutations, so you are moving very locally around it, and measuring the fitness. So we can use this data in various ways. Then there are what are called in vitro evolution experiments. So in vitro evolution works in this way. You start from a sequence, for example, 1-beta-lactamase. You take the sequence and you take the DNA. You do PCR, so you amplify the DNA, but with a polymerase that makes mistakes. So you amplify the DNA, but you introduce mutations, typically 1 or 2 mutations. So you get a library of mutants that have 1 or 2 mutations. Now you take this DNA, you put it in plasmids, you put the plasmids in bacteria, you put the bacteria in culture with antibiotics, and you see which ones survive. So now you have mutants that have survived, so they are good, and you take out the DNA and you do it again. And you keep doing it until you run out of money or time. And so in these experiments, typically they can follow, and this is done in very controlled conditions. So you know exactly how much antibiotics you are putting and everything. And so you have an evolution experiment. The problem is that it's very slow. It takes a lot of time. So you can do, in these experiments, they did about 20 generations. There are more recent experiments that can reach 100 generations, and even more recent experiments where they found ways to do this without having to take out and put back the DNA at each step, so they can do it online. And so they can reach 500 generations, and maybe in the next years we will be able to reach 1,000 or 10,000 generations. We can dream that we will have better and better data. So these are experiments where you start from a wild type, a natural sequence, and you look at how it diversifies in very controlled conditions. You typically reach a sequence divergence that is much smaller than nature, because the time scales are very short. So you typically can mutate 10–20% of the protein, but sequencing is not very expensive, so you can sequence a lot, and you can have many sequences at the end of the experiment that you can read. The last kind of data, and then I stop, is data where people choose 2 natural sequences that are at a certain distance, a certain number of mutations, and then they look at paths between them. So for example, in this experiment from a few years ago, they had 2 proteins that are, one is fluorescent in the blue and the other is fluorescent in the red, and they differ by 13 mutations, and they did all the possible intermediates. So you have 2 to the 13 intermediates where you take the 2 proteins and you mutate each possible site. And so they tested the fluorescence of all of the 2 to the 13 intermediates, and they found this bottleneck shape where you have blue proteins here, red here, and then in between you have a switch of fluorescence. So you can look at the space connecting 2 proteins, and you can do similar things by reconstructing ancestral states. So you take 2 proteins, you reconstruct an ancestor, and then you test sequences along the path that connects you to the ancestor. And so there are more and more experiments that also give you paths connecting different points. So the idea is to try to use this data or to model this data by what I said up to this point. So that's the goal. Okay. So tomorrow, to conclude the discussion, we will try to make sense of this data using the kind of models I introduced.
Okay, thank you. Thank you, Francesco. Very good. So this evening, we have this JSTAT dinner.